I want to try to end the summer school with what you may call in Latin a magnus opus, like one subject where the tools, the intellectual tools, the methodological tools are being all used for one purpose, and where also one sees some of the triumphs of the computational and theoretical approach in biology. And it is very fitting that I speak about this here. It's an NSF summer school, because the subject that I choose is photosynthesis mainly, even though I put it in the somewhat bigger context. But photosynthesis is one of these things that nobody would dare to say it's not important. I think almost 97% of all the energy that the biosphere requires for its survival on Earth comes from photosynthetic processes. And so we all depend on it, but it doesn't belong to NIH, where all the money is. And agriculture is not really a theoretical activity, even though they actually do a lot of theory nowadays to develop new pesticides and so on. Even some of the work here is related to that. But it is actually the National Science Foundation that is interested in the basic science, no matter what it is, as long as it is interesting and relevant. And so they actually are the funding source for this kind of work that is still very important for the life sciences in a broad sense and not in the medical sense. So I'm speaking now about the molecular machines of the living cell, and I just choose the photosynthetic system for the reason that it's really a wonderful example. And I will tell you in a moment why and what I mean by that. But as always, when I have to give a lecture, I have to refer to the people who I had the privilege to work with. And I have to take somewhat dated photograph of the group, because some of the people that have been involved are actually not around anymore. They have now faculty positions, postdoc positions, and so they are gone. Ladies first, this is a work of Anna Damjanovic, who is now in Baltimore and had to take a year off raising her son, and will now soon be joining academic world as a faculty member. Thorsten Ritz, they both were graduate students and the work was done, is now a professor, assistant professor at physics in University of California, Irvine. Johan Kostin you met, he gave the second day's lecture, and you can see here, and you have heard the voice of Meli Sena, who is sort of currently involved in it. And there are some other people that I will mention, Sen-Yung Park, Dejulu, and Ulrich Kleine-Katthöfer as I go along. They are also still around, and you might have had an opportunity to meet them. So, but this is, I do not try to give it as a research lecture to impress you with all we did. In fact, I pared it down. I'm pretty sure that some of my young colleagues are a little bit disappointed that I don't show everything and don't show you what they called in the Spanish Inquisition the instruments, the real tough equations. I give you a little feeling for it, that it's really elegant theoretic theory that is being done here. But I want to actually make it a lecture that we have a chance to get through and comprehend, so that you're getting the key ideas of how you actually begin to have a much more integral approach to, in biophysics. In the old days, you just took a protein, and even a protein, you looked at one process, you were very proud to let it wiggle and say, oh, this one step out of 20 steps this protein is doing, I understand well. Oh, don't ask me where the protein is in the cell and what the function is, and if what I have to tell you is really relevant for the overall picture. Now, one of the reasons why we can address these kind of more integral questions is actually very trivial, and that is that the computers that we can afford ourselves today and that the centers give us, the National Science Foundation centers, are becoming more and more powerful, and so we can do bigger systems. And many people, wonder how stupid I am that I drive all my coworkers to do these large simulations. Like, oh, Schulten wants to do now 400, 500,000 atoms. I'm really not interested in the number. But what I'm interested in is that nature is different from innate matter. And a key difference is that in nature you have many components that come together, that organize themselves. An individual enzyme is not any more interesting from a rational point of view than a catalytic surface. Both of them catalyze a reaction step. But it is that you just get the right number of enzymes together to make a whole biosynthetic sequence, a metabolic pathway. That is really what is the miracle of life, what really is the essence of life. And now by going to bigger systems, we can now study how actually many of these proteins not work individually, but how they work together, how they carry out an integral function. And in a way, we become engineers. An engineer, talking, let's say, with a physicist about building a bridge, might encounter the physicist telling him, oh, this cable there, I got this material really very, very strong. It's 100 times stronger than every time before. And it will not impress the engineer so much, because the engineer has to be sure that the function of the bridge works out, that so many cars with such a weight can drive over the bridge for the next 50 years. And many factors contribute to that. That one cable is very strong, but some other cable is too weak, would make it very bad. So the engineer has to go through the whole design of the bridge and has to find the weakest point. And that's sort of what and that might be the great breakthrough in bridge building, that you find one weakest point could be strengthened again, made to work better, so we could build an even better bridge. And so that is how we approach the cell. We look at the cellular function in an integral way. This is what the cell wants to achieve. And then we look how it really gets there. And we want to understand if all the key steps that are necessary are really make sense to us, or if there's something that is still a wide open gap. And so this is what I mean by this, and this is why going to larger systems is really very natural for living systems. There's nothing more natural at the moment, because we want to understand how things organize together, how proteins are aggregating, how they repair themselves, how they function together. So just the history here would be that, BPTI became possible actually here at NCSA because NSF gave money for, I think, 1985, 86, for one of the first supercomputer centers running a Cray 1. This one here was impossible, not quite the Cray 2, but the following machines, like for example, we run this system on one of the first clusters of Hewlett-Packard workstations. So I think this cost about $150,000 of workstation. We put them together, we could run it. And then this one here becomes possible today sort of routinely with this wonderful computer Lemieux at Pittsburgh Supercomputer Center. But there's some smartness in going big. It's just not big for its own sake, but it's big for the sake of understanding the very mystery of life, how systems organize, and that means you have to go to bigger scale. So here is sort of little more systematic statement in this regard. So here we have a cell. In the cell we have various organelles that carry out the function of the cell. I don't want to go through the whole cell. Here we have an animal cell and here we have a plant cell. And we see, for example, the nucleus that contains, that stores the genetic material. That means you have to have a library where you can put the books very dense, but if you need access to the books, you must be able to move the shelf quickly into the open to get to that book from where you have to read the protein that you want to express now. Then you have to regulate this information to be sure that the right things are expressed at the right time. And then, of course, you must then also, in the more peripheral systems outside the nucleus, synthesize the proteins. You need a machine for that. You need also a machine for the energetic needs of the cell that are, for example, met by mitochondria that turn foodstuff into the energy sources that drive the metabolism and other functions of the cell. You have many signaling pathways that you need to understand. You have cell motion. And so basically you may say one of these key functions in the cell, let's say cell motion, or let's say, as we will say, photosynthesis. How does this actually work? And I would like you say, and maybe you take a theme for your research, for your life, is better than just looking for one protein. That was a great thing to do 20, 30 years ago, that you were hitting on the right protein and could make a career with it. I think you can only make a career if you choose an interesting cell function and try to understand that, and many components will play a role there. And so what I mean by one of these functions is then shown in the first example I want to tell you, and it's not an animal cell or plant cell, as you know from you're an expert in being taught, that you teach best simple cases. And so we try to find a photosynthetic system that shows all the key aspects of it, but is the simplest case possible. And so we go to a photosynthetic organism which turns out to be the poorest of the poor. And that I mean by in terms of light of photosynthesis, you have clearly a hierarchy of organisms because you need sunlight. And those that are the rich guys on the block are the vegetation, the plants, because they are most exposed to the sunlight, like the big trees. They take the best of it. The underbrush gets already less. And now there are some bacteria that live actually in the topsoil, little bit, just a few millimeters below where nothing else is growing anymore. There are some bacteria running with photosynthesis, or on the bottom of the pond where all the algae have already harvested away all the other photons. So these are bacteria that have to make a living with photons that are not being harvested already by these other organisms. These are photons of 800 and 500 nanometer. So this poor little chap is called purple bacterium, is what we want to look at. If you are looking at the purple bacterium under the microscope, you see it looks like this, and these little membranes inside the cell look immediately very conspicuous because they're green, and they are apparently full of the green stuff that we also know from the plants, namely chlorophyll. And now if we look at one of those membranes, then we come to the machine that has one of these integral functions. And this is a machine that we will be studying for this lecture and the next two lectures. It's called the photosynthetic apparatus, and we sometimes call it PSU, photosynthetic unit. And you see it absorbs light. It's a machinery of several proteins. It absorbs sunlight, and it synthesizes ATP at the end. ATP is a very important fuel for many processes in the cell. And in fact, each of you synthesizes a lot of it every day. And you might be very surprised to know how much ATP you are synthesizing today. You are synthesizing today, depends a little bit of if you move, if you sleep now, maybe a little less, if you're active, a little more, your body weight. this is how much you need. Of course, you use it immediately, but that is how much on the overall you need. And so this is very important stuff. And so this is a key machine, and we're getting the driving energy from food sources, and but eventually we get the food sources from, we may eat a steak, but the cow eats grass. Eventually we're getting from photosynthetic organisms. And so this is a very essential machine for all life on Earth. It starts with a machinery that just is absorbing as much photons as possible from the sunlight, because these poor bacteria live in pretty dim light conditions. They have to really catch every photon there is, and so they're surrounding what is really a solar battery, a biological equivalent of a solar battery, the reaction center. They're surrounded by many proteins that have the main function of containing the chromophores that absorb the 800 nanometer and the 500 nanometer photons, and lots of them, 200 to 300 chlorophylls, and they absorb more the red, the 800 nanometer, and the 500 nanometer is absorbed by carotenoids. They have about 100 of them. So they absorb the light, they are getting electronically excited, the electronic excitations then migrating to the reaction center, and the reaction center is nothing but a solar cell that, just like a solar cell at the p-n junction, if you maybe know this from your classes, uses energy of a photon to drive an electron from one side of a diodic element to the other side. And so here our diodic element is a membrane, and the electron is being moved over to the other side. So we have a negative charge here, positive charge here. This way we can charge the membrane and put energy into it. But you know yourself that some energy sources you cannot keep them well. You might not even want to have them, too close to you. Like, you drive your car with gasoline, but you don't want to store it under your bed. And so there are energy sources that keep well, and others that are very flammable and can destruct. And an electron shift is one of those energy sources that is not explosive maybe, but the electron has a high probability of conducting back, and so you cannot have that electron separated for any long time, longer than a millisecond. You must immediately put it somewhere where it is caught, where it's caught. And you do that by taking a molecule called quinone, and you put one electron on the quinone, then it becomes unfortunately a molecule that has now one electron more than usual, and that is usually called a radical by a chemist, so it's a little dangerous molecule. And you quickly have to put a second electron on the quinone, that you have a pair of electrons there, then it's much more benign chemically and not chemically aggressive. And so that's what the system does. But to neutralize the system, it adds two protons, and so from the side, and so you really have then a QH2 molecule formed. But you will remember that now we have two positive charges here, and so this cannot go forever. We would have, next time we would have 4 positive charges, 6, so we have to replenish electrons here. So we need some kind of circle, and this is shown here by this blue light. So a proton red and an electron blue goes onto the quinone, turns it into hydroquinone, QH2, and then comes another molecule called the BC1 complex that shaves now the electrons off the quinone again and puts them back with a molecule, a very famous molecule, a cytochrome, that shuttles electrons in many processes like this. By the way, our processing of food molecules turns it into energy very much also depends on very similar so-called redox reactions, namely electron transfer reactions. So the electron is being put back, and now, however, the energy that was in the form of an electron gradient is now made into the form of a proton gradient, because as the electron goes here, the two protons that were associated with it are being put to the other side of the membrane. So now we had first a very feeble storage of energy in terms of an electron, they can tunnel too easily back, so then the cell turns into a proton gradient. That can be stored much better, minutes, and is much safer because the protons cannot tunnel easily back through the membrane. But proton gradients can still leak through the membrane, and so you must now take that energy and put it into even longer storage. And that is what is done by this very marvelous molecule that synthesizes in this bacterium and also in our bodies. It's a very ancient invention, this protein. It synthesizes ATP by utilizing the proton gradient that drives the synthesis of ATP. And so now you went through it all, and you saw that we really have here a cyclic process of electrons, a cyclic process of protons, and we just consume photon energy, and we are storing it into synthesizing from ATP, and of course now we have a cycle return to ADP by those processes in the cell, like in our muscles, and in many synthetic steps, where the ATP is then turned back into ADP, giving off its energy into the various reactions in the cell. One of the greatest miracles how that works, even though it's such a ubiquitous fuel in cells, we do not understand yet in any case well why ATP is chosen for this purpose, why ATP is so fabulously able of releasing its energy into a meaningful way, coupling it to all kinds of processes in the cell. And this is really one of the big questions of your generation to answer the mystery of ATP, why nature chose this molecule over many others, and why this is such a wonderful, capable molecule of releasing its energy and straight coupling it to something where it's used rather than exploding and wasting most of it somewhere else. Okay, so now the new biology or the new biophysics is to say, I want to understand this system. I do not want to understand one of these little pieces of one of these proteins here, but I would like to really develop a complete understanding. Now that is of course easy said, and it's a hard goal, because you have to now really go and first build all the systems together. So even so you say, I want to understand the whole thing, doesn't mean that now you just write some integral equations or do some philosophy. You still have to do the hard work that previous generations did, maybe even more so. But of course you have better tools. You have the computer, you have lots of structural information, and so you can do more. It's hard work, but you can do more because we are now 50 years later and there are more tools available. So you have to go through an enormous pace, like, you know, people said in the old days, I don't want to, but maybe I'll tell you this story. It's sort of like a cute story, you know, it takes now 2-3 minutes. So in the old days, people thought in biology that you cannot be possibly serious if you work on more than one protein. So that was just a giveaway that you are really awful. So one day I decided, we got into these motors, I wanted to work with Ken Holmes, he's director of the Max Planck Institute in Heidelberg, a very famous muscle protein expert. And it was very difficult. It took me about a year or more to woo him, you know, to say, oh, I'm really interested, and we did a little bit, and finally he said, okay, Klaus, you can come. I knew him actually pretty well from skiing, so, you know, he was sort of like a buddy of mine, but it took a while. And so then he finally invited me, and I went to his institute, and we talked, and it went well in the morning. We went for lunch, and then I met all the co-workers, and it went really well. And so I thought already that I would go into work on myosin with Ken Holmes, a very famous protein. And then suddenly, at about 4 o'clock, the whole atmosphere became very melodramatic. like almost like a church wedding, sort of. He said, please, Klaus, come now to our group room in our Max Planck Institute. And people were sitting there with their hands folded around the table, said, please sit down here. And then he said to me, Klaus, I would like to work with you on myosin, but you must now swear that you will only work from now on myosin and on no other protein. And I'm more a philanderer when it comes to proteins, so I knew I couldn't satisfy, and I had to just leave, and that was the end of the collaboration. So that was 50, actually that was not even, how you see, you recognize some people here, I hope. That was some time ago. Today you have to work on many proteins, so we really have to make it, but you should remember those were the great founders of the field of protein biophysics, and they had a point, and we have to take every protein very seriously. Okay, so we have to first, we want to analyze the whole thing, and we will go through the whole thing, but we need to still work from the detail up in order to really put our thinking on solid foundations. And so here we start. We start first with what is really the central part of the initial step in this machinery, namely one of these round proteins surrounding the reaction center. Now actually I want to make it easy for you. I want to win you over. The hard work involved in this, actually some of it done by Felix here, I don't want to share with you at this point. Later I will not spare you the details. But this moment we just would like to say, okay, let's have a close look at this system in order to appreciate what is really the issue here. And so you see here that we have a circular protein. We have the reaction center. The reaction center, a very famous protein. The crystallographers who solved the structure got the Nobel Prize for it. And so it was one of the first membrane proteins completely solved. And so here's another membrane protein then surrounding it. You see the green stuff there, that's some chlorophyll. And we want to look inside, and we did it doing our molecular dynamics. And I made some movies, just like you learned. I learned actually also from the wonderful tutorials people wrote, so I made a series of movies here. So here you see a box of water. Inside the lipids, the yellow are the lipids, here this one. And then the reddish on the front and top is the water, and inside is the protein. Now we take everything away except the lipids. Now you see the lipids here. You see that the lipids have the shape of this box, so of a square or a hexagonal shape to make it have a minimum amount of molecules and still periodic system. It's better than a square box. And so you see the lipids here. So we don't show the water on top and bottom. We don't show the protein. You see that there are even some lipids inside because we have a ring molecule here, and then we have the reaction center, and obviously there fit some lipids inside the ring. So the ring forms like a courtyard, and in the courtyard is the reaction center, but also a few lipids. So now we add the protein. And so now you see that the protein fits in there, and you see that there's a circular protein containing the chromophores, and then the reaction center that is a solar cell. And now we just take the lipid away. We just see now the protein. And so you see the reaction center inside the circular protein. And now comes the big step. The big step is that we now add all those prosthetic groups, chromophores, that are actually functionally important for the absorption of photons, for the excitation migration, and for the solar cell function of the reaction center. So here you see them, and so you see they are packed inside there. You see really, look at this beautiful thing, how this ring molecule is really sort of two narrow fences and fully packed with chlorophylls. So you really see very quickly how the system is really designed for its function. You know, the system lives in dark in dark territory, and so you really want to have as many molecules as possible that absorb the light. And then you see that we need to understand how the excitation is transferred to the center, where the molecules are located that are involved in the function of the solar battery, of the electron transfer. And so we have a feeling a little bit that what the proteins are doing here is they're a scaffold that just hold this chromophores. That makes sense because, you know, when you move an electron, you don't have to move a protein around. You just keep the components, the electronic components together. So what we do is now we just take the protein away, and now we have here these groups, and now these groups are the ones we need to understand. This aggregate, this hierarchical aggregate of molecules is really the reason why this all is. And if you want to have an understanding of it, then we have to understand the physics of these systems. Possibly also the motional characteristics, because as we will see, actually this system works like a Swiss watch, but with all the parts rattling around. You know, that's nature. Nature built all these systems at physiological temperatures, and everything is rattling and still functions well. In fact, nature very often uses even the thermal motion to its advantage to make these Swiss watches work better. And since it's such an important component, we blow it up a little bit, and now we have it in full beauty. And so this is basically what we need to understand. These molecules, they are the ring molecules here you see are chlorophylls. This is a heme molecule. That is the ring with an ion in is called a heme. The ring with the magnesium in the center is called a chlorophyll. A little bit different side groups. And then we have the quinones that I mentioned. Here are two quinones that are involved in this, putting electrons and protons on to turn to form hydroquinone. So this ring absorbs the light, and the reaction center here in the center is in charge of the electron transfer that charges the membrane. And I want to start with looking at this process. And so I take the ring away, even though its a primary function is to absorb the light, and then the transfer excitation to the center so that the energy is then being utilized for the vectorial transfer of electrons to charge the membrane. And so we just take now the center things, and now I wobble it a little bit so that it doesn't rotate away from my side, so I can talk about it a little better. So it is this pair of chlorophylls here that actually is at the real center of it all. And these pairs of chlorophylls get the excitation energy from all the other chlorophylls, the 200, 300 chlorophylls in the system, and the carotenoids. When they get excited, they send the electron excitation to the thing. This then uses the excitation energy to excite one of its electrons and conduct it through another chlorophyll that is here to pheophytin, that is just like the electron, like a chlorophyll, except it has no central atom. Chlorophyll has magnesium, pheophytin has nothing. So as a result, the electron gets electronically excited in the chlorophyll, goes to the pheophytin, is there at higher energy. And now this electron is now being conducted through this quinone and an ion atom to this quinone. And this quinone here can shuttle in and out. And this is actually where herbicides act, like Roundup. You are designing a herbicide that has a shape of this quinone, plugs for particular weeds, this functional position, and so as a result, this very, very important process doesn't work anymore. So this is how it goes. Electron gets excited, conducts through these steps, comes here, but now we have the hole here, one hole, and now we need to fill it. And here we have a heme group that is actually not belonging to the reaction center, but that is already a cytochrome heme. There is a cytochrome sitting here that is bringing the electron from the remaining part of the apparatus. So this heme comes with an electron, fills that electron, and then the system can send a second electron. Another heme comes, fills the electron again. And so here you see this is really the conduction pathway, the wiring of this system. And so you see that even though we start big, we are really now very quickly focusing down to the heart of the matter. So we are not, the shallow big guys who want to understand, because we cannot understand one protein, we rather than prefer to understand 20 proteins. No. We have to be intellectually honest. We have to see where does it matter, where are the key processes, and very quickly, even though we want to understand the whole system, we are narrowing down where the key action is, here, there, to put it then back into the whole picture. So there's no cheapness in doing integral biophysics. It is actually harder. You have to also go do the detail, but you do the detail that matters, and not only the detail that you can do. And so here we are now at the detail that matters. Now, when it's a control, this electron transfer rates, one wants the electrons to go forward and not backward. I mentioned the danger of the electron going backward. And so you need to control the electron transfer rates. And so in a computer, you might have a copper wire, and so everything is homogeneous. But here we have very heterogeneous material. So you can imagine that you can control this rate from every step to every step. And so, in fact, you can. And the question is, how is this done? Now, certainly, one factor is the connectivity of these molecules, how close they are so that the electron can tunnel from one to the next. But very, very important is that the electron carries a charge. All the atoms of the proteins also carry partial charges, as you saw in the parameter file and the structure files. And so the electron that is being conducted interacts with all these charged atoms that are carrying out vibrations. And so in order to understand the regulation of electron flow, you need to keep the protein matrix in mind, and even actually the membrane and the water surrounding it. Because the electron has a long-range interaction. Here we see this heme group, the two chlorophylls, the conduction pathway here, the iron atom. And now I show superimposed here the volume of the protein, because this matters. This is a key part of the control of the electron transfer. And that is what I want to discuss with you. I could have also discussed the quantum chemistry of the electron tunneling between these units, but I just choose to select this one to give you a flavor of the field. So, I cannot be completely here, so there are other aspects that I cannot mention now, but I want to mention this aspect. And I want to mention it because it really is a wonderful example to see how you can approach a question in a very rational, intelligent way. So I say I want to understand how the electron flow is coupled to this, to the vibrational motion there. How good, that's a good question to ask, but it's sort of like a fuzzy question. And you would like to ask this question as focused as possible, so that you really know what kind of properties of the protein matters in terms of controlling the electron flow. And that requires thinking, that requires actually borrowing from the condensed matter physicists who have been dealing with electron flow in crystals for, now over 50 years. Actually, I think now it's sort of 70 years. And so they have a lot of notions. They deal with crystals. They don't deal with soft materials. And so you have to be careful in dealing with them. But on the other side, there are people who are intelligent and who have been dealing with the matter, and so that's a possibility to borrow from them. And that you can do that, I want to show you now. So here we come back to our overview, and here we have now the reaction center, and we want to understand how the electrons go through the reaction center. So I moved it now to this side. Here, these prosthetic groups, and now we see that we want to now see that electron. So it gets in little, this corner is not easy. You see that electron, that arrow there. So this is the first electron transfer step. The second, the third, here's the quinone. Here, at the moment the site is empty, this is where it's shuttled in and out. And these are the electron transfer steps we need to understand. Of course, also the electrons are coming back from the cytochrome to the heme group here. So this is, we need to understand these steps. And we will pick one step, let's say, for example, this step, how we go from this quinone to that quinone for our study. Let's say we want to understand it. So now we would like to know how this particular step here is coupled to the motion of the protein. And so, the electron can be either this quinone or that quinone. And now the electron has a negative charge. All the atoms in the protein have a partial charge. The Coulomb interaction is long range. And so basically you can imagine, at least for your thinking now, it's a good picture, that each of these atoms is carrying out a vibration. And now there's an electron, the atom is partially charged, and now the vibration depends on if the electron is here or there. Because if it's here, let's say it's a negatively charged atom, then it might be happier, or maybe moving more around this point, whereas when the electron comes closer to it, it might very slightly displace because the electron pushes against it with a repulsive Coulomb interaction, whereas another atom here might be attracted because it has a positive charge, so it might move on the average towards this group when the electron comes closer. And so you have sort of two pictures here. One is the electron, one is the vibrational motion of the protein before the electron moved. And let's say we define all the positions as 0. So don't worry that, like we can differentiate between them, but let's say just we write it like this. And we have a harmonic oscillator for all the atoms, kinetic energy plus quadratic potential energy. And after the transfer, we are getting a shift, a positive or negative shift depending on the sign of the Coulomb interaction. For all the atoms, we sum here over all the atoms of the system. And so this is, of course, a very diligent kind of work, but you ask yourself, is this guy crazy? Because we have in this protein about 10,000 atoms. And so we have 30,000 oscillators in the X, Y, Z direction. And each oscillator is characterized through a frequency, a mass, and a shift. The shift is here sort of written down as a coupling constant, sort of like this mass. So this is, we call it a coupling constant. And so, other than calling it a shift. But so we have then 90,000 constants describing this coupling. And so it's, a good approach. And actually some people, actually here some delegation from Duke University, from the Beratan group, and they would probably say, oh my God, this is even too easy. You have to do more and better than what you are telling us now. But this is actually sort of like the first and simplest picture that you can have, but you are confronted now with 90,000 constants to describe it. Okay, but theoreticians are sometimes crazy and they forget about reality and they go on. And now you can go actually and express the rate of electron transfer for the electron being coupled to all this vibrational motion of the protein through a very succinct expression that's written here, an integral, where you have here just the energy difference that it requires to go from here to there, and then you have actually the average energy difference, and then you have here two functions, and these two functions depend on all these horrible parameters. You can actually scale away the mass, so you can put it into the frequency. The mass is really not an independent parameter. You can say actually you have only 60,000 independent parameters, coupling constant and frequency for each oscillator. So, okay, succinct expression, but still horrible amount of information necessary to do something with it. And now comes these beastly intelligent physicists who did the following trick. They say, let's write these two functions as an integral over what they call a spectral density that contains all this information on the frequencies and on the coupling constants, and then they write Q1 and Q2 just as these integrals over these densities. So by the way, this is actually a wonderful example of the famous Feynman path integral method. So if you read Feynman-Hibbs, that's that old famous book, you can actually nicely understand the derivation of this formula, even though it's very elegant to use every trick in the book, but you can make it. So you can write now this expression. If you then summarize it a little bit, then you can say this rate of electron transfer can be written through these two functions, and the functions all contain only one piece of information, namely the spectral density. But the spectral density depends on 60,000 parameters, but now you making a wonderful discovery that actually the people also realized in the condensed matter situation, that the spectral density actually can be measured independently, either experimentally or through simulation. And you can do the following calculation, simple calculation to get this spectral density. You do not have to bother about all these parameters, because this property contains these frequencies and coupling constants implicitly in a form that you need here. What you do is you calculate how much energy does it take to transfer an electron from this quinone to that quinone. Now this energy depends on the vibrations of all these atoms. So this energy is fluctuating in time, because depending on the charge environment, it's easier or harder to transfer the electron. And here you see how this energy fluctuates. At this moment, we transferred actually the energy, and then now the electron is here, and the protein sees it and continues its sojourn here. And so we look at this, we call it the energy gap function. How much energy do we need to transfer it? And we calculate this energy gap function. And now it turns out that the spectral density is given by the root mean square deviation of these fluctuations, the correlation function. If you know the correlation function, then you can calculate through a cosine transform the spectral density. And you also need the average energies before and after, because that is this E that comes there into this Fourier integral. And so you see now that in some simpler picture, but some actually very powerful picture, you see what kind of property in the protein matters to control this electron transfer. It is actually how the protein controls the energy that the electron takes to go from point 1 to point 2. And you can calculate this energy through a simulation. And once you can calculate it and get the correlation function, the root mean square deviation of it, and the energy difference, you can then calculate the rate, and you do that. You need also some quantum chemical information, but for example, for the initial rate, you find then it's a picosecond process, and you can see, for example, how it depends on temperature. Here's a very surprising result that was actually one of the intellectual engines of much of biophysics for several decades, and that is that the rate of electron transfer becomes slower as you increase the temperature. That is not what a chemist usually sees. A chemist usually sees that the rate increases when you increase temperature, because you go over a barrier, and then you go easier over a barrier if you have more energy, thermal energy available. But here we have a rate that goes down. And the reason is that this coupling I just described is more efficient when the vibration of the proteins are more coherent at low temperature. At higher temperature, they become less and less coherent, and the coupling is not so good anymore. And so it's really the coupling of to the protein that is controlling this behavior, this rate. So you're doing this too. You will be calculating your own energy gap function, and you do it for the cytochrome that is delivering the electron to the reaction center. Here you see the cytochrome, and you are being given the coordinate files and the structure file to run a simulation. And so you have to then to calculate this energy function. What you do is you have here the heme. It is negatively charged when it comes. It transfers an electron, and then it becomes neutral. And you are being given the charge distribution of the heme before the jump and after the jump. And you need to calculate now the Coulomb energy difference between the interaction of the heme with the rest of the protein when it's negative and when it is neutral. And that energy is the energy gap function. And you calculate this energy difference, and that is the input that we needed to see how the protein controls the electron transfer rate. There's, of course, also an intrinsic contribution of the redox energy of the heme group itself, and to calculate that you need to do quantum chemistry. We don't include that. We only include how the protein controls it, but the protein is a very, very important contribution going beyond the redox energy of the heme itself. And so you will be carrying out a molecular dynamics simulation of the cytochrome. It is in water. You can only do a short simulation of 500 femtoseconds in the time that is sort of like available in a tutorial session. And in order to give you some better results, what we do is we give you also a trajectory of the cytochrome that lasts 10 nanoseconds, so that you're getting better data for the energy gap fluctuation. And then you can do the statistics of it and see then that, like here's the root mean square deviation, that's the prefactor for the spectral density. It is very hard with the statistics we have here to get a good correlation function. You can try, but I think that is a little harder to get a nice one. You get, of course, the average energy contribution here, and so you see how you can, through a simulation, understand or get the information that is needed to understand how the protein controls the electron transfer. Okay, before I then end my first lecture, I would like to tell you a little bit about biology to make you appreciate that biology is a field where you cannot be narrow. You have to be very broad-minded in biology, and here is, for example, the question that we want to understand. If we have here our light harvesting system, reaction center, big ring, we will see in a moment the small rings, but now let's look at this one, that there is a gene here, it's called the PUF gene, that controls the synthesis, the expression of the protein, the reaction center. It has here this red and this yellow part, this is L and M. And then we have here this ring, and the ring is made of 16 independent units. And actually 16, but you see a little bit of color change. You can see that here a little bit. The inside is one protein 16 times, it's a A unit, and outside is another protein, it's a B unit. And so these two genes synthesize this stuff here. The B and A synthesize this, the A, this the B. But you realize we need this one time, this gene, and this one we need to express 16 times. Yeah? Because we have one reaction center, but we have 16 of these units. One here, 1, 2, 3, 4, 5, 6, if you go around 16. So how is it done? This would be pretty stupid. Now you could say, okay, then maybe what the system can do is it can make 16 of these genes. So then they're all expressed. And so if you have 16 of those and one of those, you get the right stoichiometry. But you may think maybe that's a little wasteful. You have the information once, why write it 16 times? And maybe, like one of them gets mutated, the other one doesn't, and then it could be complicated. So it seems to be better to have this information only one time, and this information one time in one gene, very good, information there, blueprint is there, but you have to say, do this 16 times. How is it doing it? Now here you see then some part where you see these fields of biology, namely on one side molecular biology, on the other side genetics, come together. And if you go to the database and look at the genetics, you find that this piece here is being expressed when the gene is expressed into a messenger RNA that contains information on this system and carries it out of the cell, of the nucleus for synthesis. And if you do it like this, you would express this one time. But now, the messenger RNA has here some pieces that are non-coding. And here you have a piece of RNA that forms, those of you who deal with RNA know this is a very common motif in RNA, that forms a clover leaf, or a little loop. And this one does as well. And now it turns out that these hairpins or protrusions are recognized by certain enzymes. The enzyme cuts this out, and then you have, and destroys this one because of this kind of information is destroyed. And so you're getting then, once in a while, the full gene, and once in a while, you're getting only this piece here. And now there's another well-known heuristic rule that the longer a messenger RNA, the shorter lived it is in the cell, because there are always processes in the cell that destroy things so that nothing hangs around too long. And so this one gets destroyed much faster because it's more exposed to these enzymes than this one. And so now you have this one living about 15 times longer than this one. And so you're getting now one of them, 15 of them, and altogether you have 16 of those and one of those. So you see how clever nature designs its system. Just here through the lifetime of the eventually coding RNA, it gets the stoichiometry right. By the way, here are some elements that are coding that we have no idea really of what it is doing. And one of them we know a little bit, but there are some genes that we do not know, and the feeling is that the genes that we do not know are very essential, but they are somehow disappearing from the stage all the time. And they are likely genes that are there to help the assembly of the machine. So if you don't have the genes, you find it actually by knocking them out. The systems don't grow well, they don't build this apparatus, but when you look at them when it's ready, they're gone. And so you have some genes, assembly genes that couldn't catch yet. Hopefully we find them one day and then can catch them on the act of doing things. But you realize here that there is also a very interesting aspect of assembling this machine to begin with. And you have to find the genes, you have to find the protein that might be helping and chaperoning this. And so this is also, one of these problems that you have to deal with. So you go all the way from structures to genetics to physics and so on. So this is my first example. This is all written nicely down in this review that is available on our website as a PDF or from the journal. And so it's a very detailed description. And you see in particular also Xiqi Hu, who was a postdoc in the group and who is now professor at Toledo, is the first author. He was actually the one in the group who did much of this work. Okay, so that was the first part. So